Hello, boys and girls. Mr. Forte again with hopefully a helpful review video on genetics. All right. Genetics, all living things have a genetic code. Basic genetics are what make us what we are. If we make a tree, if a tree makes a tree, a frog is a frog, okay? They all have this DNA. They all have DNA. They all have chromosomes, which make them what they are. Genetics are the instructions on how to make an organism. And the machines and things to create something from those instructions. I think a blueprint on the house to build a house is useless unless we have the hands, the materials, the machines, then that are going to help us read those blueprints and assemble the house, right? And organisms receives their genetics from their parents. Our parents are humans, so we turn out to be humans. An amoeba's parent was an amoeba, so it's going to be an amoeba. We're the result of sexual reproduction. So we're a mix of our parents and even our grandparents all the way down the line. This is a heredity. We inherit our genetics from our mom and dad. And sometimes, mostly, it's good stuff, right? How wonderful our eyes look, or nice hair, or our intelligence. We inherit genetics from mom and dad. Even early scientists saw this. They saw that something was passed on to the next generation. I mean, you look at it, the kids look like their parents in some way. You can see the resemblance, and kids even look like each other. But of course, they're not identical because of certain things. Remember, heredity has to do with a lot to do with what we get from mom and dad. Now, they can only donate one chromosome of their pair. So during meiosis, mommy, through random assortment made, don't donate this big B, and daddy also donates a big B for brown hair. Now, mommy could donate a big B by chance, and daddy donates a little B. This kid would have brown hair, but is a carrier for blonde hair. Here, mommy could donate the little B gene, daddy could donate the big B. And here you have another one with brown hair, but carrier for blonde. And then this last situation, mommy donates the little b, and daddy also donates the little b. And there you have a child that's going to have blonde hair, right? That's great for just talking about hair color, but sometimes mommy or daddy may send something that isn't very fun to have, like cystic fibrosis or sickle cell. Here's another one. Once again, parents send one chromosome, all right? They unite in fertilization, and that's the zygote. That's the first cell with all that new genetic material and has that uniqueness because of the mix that mommy and daddy sent. Remember, no two sperm and no two eggs are alike because of crossing over and recombination of genes during meiosis and the random assortment of chromosomes. Some particular traits that we have are just from a single gene. You have tongue rolling is, believe it or not, is a single gene. Having a curved pinky finger is a single gene. A little cleft chin, that's what Mr. Forte has. You can't see it through his beard, but I have one of those. This guy, Terrence Howard, he's an actor, has a widow's peak. Some of you have that. Dimples are a genetic trait. I guess these girls are twins. They sure look like it. Hitchhiker's thumb. Having an extra finger. These are single gene traits. Most traits that we talk about are actually a bunch of genes all over our genome from our 46 chromosomes kind of working together. But there's some like these that are single gene traits. There are also some that are not good having. Um, Once again, mommy or daddy could give you something nice like a delta 32 and you'd be immune to the plague and delta and HIV, but if you had only one, that'd be tough. The normal gene on this chromosome is CCR5. It's a receptor molecule in the white blood cell. In this case, this person has one of each. All right, so if the plague came around, they'd have what? A little bit of resistance. They may get better. HIV would delay the onset of the symptoms. And of course, you could have a situation like this where, if mommy was a carrier for cystic fibrosis and daddy was a carrier for cystic fibrosis, I mean, they have one of these genes and they sent it, you'd have a 25% chance of having a child with cystic fibrosis or sickle cell. That's not so good. You clearly don't want that being sent on. So this kind of, we need to think about, if you know cystic fibrosis or sickle cell run in your family, you should get tested. You don't want your child to be living with such a terrible disease like cystic fibrosis or sickle cell. Um, so, you know, all of our genetics are passed on by this amazing molecule called DNA. It's got this double helix shape, and that shape allows it to unravel and unzip. This ability of the model to do that allows for the replication of DNA in cell before, in mitosis, and also to create proteins. I mean, think about it. You look at the structure, it looks like it can do that. It's just like if you looked at, a steamroller. You wouldn't think that a steamroller is for racing. You would kind of infer that it is for flattening things out. I like using a nice analogy for genetics, and I use the book analogy. I'll uncover these at another time. But we can do this analogy with a bookcase in a room, all right? Because that's really all DNA is. Our DNA, all the 46 chromosomes we have in our body are just a library of instructions, sort of like a cookbook, all right? And just it's the instructions on how to make something, all right? And to get that from our parents, we need to replicate that DNA. And to replicate it, there's this idea we have to understand how it works on this DNA strand. It's fairly simple. We have these nitrogenous bases. G goes with T, and G is just guanine. T stands for thymine. Wherever there's a G, there's a T. I'm sorry. Wherever there's a G, there's a C. Wherever there's an A, there's a T. All right? So that yellow one would be a C. I put a G there. I thought I had the G. All right, let me do, an A is the green, right? No, it's the other one. Boy, Mr. Footpack's colorblind. All right. And when DNA replicates, okay, it's going to unzip and unravel. Let me show you a quick video of it. So here we see the DNA molecule, nice image of it. And it's going to unravel for us in a second. Twisted double helix. And it's actually two strands. They're two complementary strands. They're not mirror images of each other, and you'll see in a second. They are complementary. All right? This one matches up with this. Okay, so here we see a T with an A. You'll see that they're not the same. All right? It's not a mirror image. They're complementary. They fit together. Imagine you had two big ones together, how there'd be almost a bump in it. It's like a ladder. It's got to be parallel to each other. And they're showing how this is just like code in a computer. T goes with A. And then we just read this code. This code has to be read, just as we read a code in a cookbook. We're reading words. Words are just a code, and it's that cookbook is telling us what to make, and that's all this is. It's just telling us what thing we need to make. And here, once again, this is happening during mitosis or meiosis, the replication of the DNA, the replication of the chromosomes, this process. It's got to unravel and unzip, and you get two identical copies. And now they're going to show a real high-tech version of this. So here's the parent strand of DNA, and here's a new strand being created. So here's the parent strand coming in. This enzyme, okay, it's called helicase, is unzipping it. This one is reading it. As it reads an A, it puts a T. As it reads a G, it puts a C. As it reads a T, it puts an A. And here's one strand going off this way, and this one is done a little backwards, but here. Eventually, you come out with two identical strands of DNA. So the two resulting cells are going to have identical DNA. All right. Now, this is a tough one for some people to get, but every cell in our body has this library of 46 chromosomes with the thousands and thousands of recipes, basically, or instructions on there to create these things. Now, what do cells create? It's proteins. Now, the DNA or chromosomes are identical in every cell of an organism, but only certain genes are turned on to allow that particular cell to perform its function. Think, the beta cells in a pancreas are activated and express the insulin gene. If you're a nerve cell, if it was a nerve cell, it's going to activate the genes needed to produce the neurotransmitter like serotonin. Cells in the bone marrow activate the hemoglobin gene to produce red blood cells. Now, what if this was a white blood cell? You gotta think, what is something a white blood cell will produce? Well, it's going to activate a gene to produce an antibody. That's going to make a copy of RNA. And RNA is just a small piece, a copy of a small piece of the A, C, T's, and G's that are in there. And RNA is going to take that code to a little machine shop, right, called the ribosome. Okay? And that ribosome is going to read that RNA code, and it's going to produce a protein. And that particular protein, if you're white blood cell, one of the many of thousands are going to be how to make an antibody. And of course, that antibody is going to go out and fight pathogens. Right? There are many, many, many different types of proteins that we make in our body, thousands. Right? We have things like structure like keratin for our nails, our skin. Our blood needs to coagulate or clot and form scabs. Those are based on proteins. We have membrane proteins like the one we saw in the video about the Black Plague. The CCR5 is a protein found on the outside or on the surface of the membrane. All the thousands of enzymes in our body. We have hormones like human growth hormone, estrogen, testosterone, receptors that are on cells so cells can communicate, antibodies. The neurotransmitter, I said, insulin is a hormone, but it's a protein produced by the beta cells. Hemoglobin, all right? All made from a code. A code had to be used to make them, all right? Now, I'm going to move ahead a little bit, and we're going to see how this actually works, okay? And we had 1835. So that was it. So here's what happens during protein synthesis. So what happens here, this is the DNA, and this is another big old enzyme reading the DNA. This is all happening in the nucleus, okay? That's where the DNA is. DNA is in the nucleus. It's not outside the cell. It's not in the cytoplasm. It's in the nucleus. It's reading that DNA, and this strip, this copy, this single strand is being copied. So one strand is being read, and it's producing this RNA. All right? And now that RNA has got to go somewhere. It's got to go out to the ribosome. I didn't show that little part there. I wanted to see that. So here it is. It's going to leave, and it's going to make its way out of the nucleus. There it is. Here it is coming out of the nucleus. Whee! There it goes. It's going to go to the ribosome. And it's going to start reading that. So here it is. Here's the ribosome, showing inside of the ribosome. So you see the RNA going through, all right? And you see these little transfer molecules. They're just bringing in amino acids. So this code is being read. 3 letters at a time are being read. And every 3 letters corresponds to a particular amino acid that's going to be added to the chain. So there it is. Boom, boom, boom. It's bringing the amino. You see the protein chain building up, building up. Okay, that's going to, and now it's folding. Right? Proteins aren't long little strands, but they fold up according to the amino acid they're next to. And eventually, there it goes, bringing in, and one after the other, boom, boom, boom. 3 letters at a time. It's reading 3 letters here. Every 3 letters means it's a particular amino acid. And there's our protein, and it's almost done. And it folds up, boom, and it's going to go off somewhere and be packaged and assembled with some other ones, and you're going to have a protein that can do a job. All right. Now, how this actually happens, we need to understand this code a little bit better. Okay. So here's a DNA sequence or a gene, a strip of DNA. It's all code. Okay. It looks like nothing, but it has meaning behind it. So TAC. All right. So in the nucleus, right? Remember, this is in the nucleus. This is going to be, all right, an enzyme is going to read the sequence of the, all right, the DNA gets opened up. It's going to read the sequence of the DNA and an RNA and create an RNA message. This is called transcription. We're going to turn DNA into RNA. And this occurs in the nucleus. So wherever there's a T, there's an A. except in this case, there's no T in RNA, so we replace it with a U. So when there's an A, there's a U. Where there's a C, there's a G. Okay, C, a G, a C, a G, a C, a G. Okay, so there's a T becomes a becomes a U, C becomes a G. All right, so now we have this RNA strip. This RNA strip, like we just saw in the video, is going to go out to the ribosome, where it's going to get translated. All right, AUG gets translated into MET, which is an amino acid called methionine. Now we look at this little code chart over here, and we look at those first three letters, which are AUG. We do it this way. Here's the A. Here's our first position, okay? So here's A. We want to identify that. So that means it's got to be in this row here. Now our second letter is U, right? So here's U. So it's got to be in this box. A, U, and here's a third one, G. So that tells us methionine is the first one, okay? Now, our second, we read, so our second set is CCC in the DNA, right? That transcribes into GGG in the RNA. GGG in the RNA, okay, is going to translate at the ribosome. We look at it. Let's go to G. Here's our first one, G. That means it's in this row. Okay? G, G, G. So here's our second one. This is an easy one. Okay? G, G, G can be, all right, it's got to be in this box. Well, clearly they're all glycine. All right? And there's the third one. So it's really this one. So our second amino acid in this chain would be glycine. All right. Now, sometimes we have a mutation. And a mutation means a change in the DNA sequence. So let's focus here on this triplet code in the DNA. Imagine during mitosis or meiosis, there was a mistake made. Remember, there's 3.2 billion base pairs in your genome. So a mistake can happen. And if it happens in meiosis, that's a problem, because that's going on to possibly be an offspring. So imagine there was a mistake, and there's an A. All right? That T becomes an A. That means that DNA is now going to transcribe into not GAG, because A doesn't become A turns into U in the RNA. Now, that RNA is now going to go out to the ribosome, and the ribosome doesn't know any better. It's just going to read GUG. And if we go to GUG, there's G, second letter U, third letter is G, okay? And that's going to be not that, but that. And every amino acid is different. You change one amino acid, or you change one letter in the DNA, that changes a letter in the RNA, and that changes the amino acid that goes there, and that can change the shape of that protein. Now, if it changes it enough and it doesn't function, that's a big problem. Most illnesses are based on mutations. Now, we look at something like cystic fibrosis. It's based on a protein in the membrane of a cell here, okay? That mutated gene creates a mutated protein, and this ion channel, okay, that is supposed to let stuff in and out of the cell doesn't work very well. And the cells in the respiratory tract and in the digestive tract become very thick. That's a big symptom when someone has cystic fibrosis. Sickle cell. These are supposed to be free-floating hemoglobin molecules. They're not supposed to be sticking together. All right. If they stick together, clear inside the red blood cells, you get this situation where we get these crescent-shaped or sickle-shaped cells, and clearly the symptoms there we talked about before, how they can clog up various blood vessels in the body and be very painful, stopping those tissue from getting oxygen and things it needs. You know, mutations that cause disease like sickle cell and cystic fibrosis occurred thousands of years ago. It's tough for people to figure this one out, if not millions of years ago, during the DNA replication of one of our ancestors during meiotic cell division. All right, and it's been passed on or inherited through the years. Because it happened in meiosis, it was passed on to the next generation by a sperm or egg. Now, for these diseases, people would need two copies of the mutated gene. So maybe many years later, two people who had that mutated version got together, had kids, and they happened to send that sperm or egg, which had that. The two people got together, and that kid probably had cystic fibrosis. Probably didn't live very long if this happened thousands of years ago, or sickle cell. Once again, not all mutations are bad. Blue eyes is believed to be a mutation that appeared in the human genome about 10,000 years ago. Delta 32 appeared about 700 years ago and was beneficial in resisting the plague. Now, if the mutation occurs in the DNA of a body cell or the skin or liver, pancreas, or lung, it could lead to cancer in that person, but that mutation is not going to be passed on. I mean, think about it. You're out in the sun, you get a mutation in here. UV lights cause a mistake in replication in some of your skin cells. The person gets skin cancer. Well, that's not going to pass it on, but clearly if they get it in the reproductive area, right, and you spend a sperm that's got a mutation and it fertilizes an egg, well, that egg is going to have that mutation. That child's going to have that mutation, okay? Most times mutations are bad, but once in a while they're beneficial, all right? Once again, the mutations have to occur in the sperm or the egg, the creation of the sperm or the egg, for it to be passed on to the next generation. Our genes are either turned on or what we call expressed because of many factors. And here you see I love this version of the bunny, right? The colder areas of the bunny, the Himalayan bunny, they turn black. And there's a reason for it. Supposedly the black absorbs more heat. This little bunny is in the Himalayas, so, ears and things, the more sun they can absorb or heat from the sun in those colder areas, I guess helps them out, all right? Cute little harp seal, right? This guy, when they're babies, they're white. And then when they get older, they look like mom or dad. Well, they're white because they can't swim yet. Most of their time is on the ice or snow, and it's going to protect them from predators. When finally get older and they have this mottled look, M-O-T-T-L-E, I think is the way you spell it, all right? They blend in better and they're swimming around. It's hard to see them from below if you were a shark or something like that, or a killer whale or something. And it helps them blend in when they swim more. So those genes are turned on or off because of the environment. Here you got the fox in the winter. In the colder months, it's light colored fur. As it warms up, it actually turns this browner color or whatever color that is. Remember, Mr. Forte is colorblind. All right. Factors that affect gene expression: the temperature, sunlight. Think about it in the summer when you leave something on the lawn, the grass turns yellow. Sunlight affects the expression of the chlorophyll genes in plants. The pH, the cells around an environment. If you took a stem cell and put it in with other heart cells, cardiac cells, guess what? That stem cell will probably become a heart cell. The food we eat, the nutrients, okay? You eat a lot of crummy food, that's going to cause you to be maybe overweight. Pathogens can affect, believe it or not, our expression of our genes. You have identical twins. They could be, every single cell in their body her body and her body are identical to each other. And yet we see twins that, after many years, look different. I mean, this one twin could end up living in the American Southwest and be very, very tan, or live somewhere else and have blonde hair. One can get cancer, one cannot. And they have different personalities too. The way their voices, anything, certain things, okay? That's because of gene expression. They may have identical genes, but they may express them differently. Now, think about it. If Mr. Forte was to start working out and taking steroids and lifting some serious weight, drinking giant protein shakes and became huge, would my kids turn out that way? No. Okay, I may have had something that helped me get really big, but that's not going to be that way. All right, I'd be a great basketball player because I practice. My kids could be terrible at basketball, or it may have a lot to do with my physicality, what I had about me physically. That's possible. All right. In this day and age, you hear a lot about genetic engineering. And basically what that is taking a gene from one organism and implanting it in another. And you think, why would we do that? Well, we can take a human gene. Think about it. People who don't produce insulin, people who are diabetic, they don't produce insulin. So we need to get insulin. They need to take insulin. They have to have it injected into their blood when they have meal to lower their blood sugar. Well, we've created, how else are we going to get it? Pigs, cows? Well, eventually that's a foreign substance. And the body becomes resistant to it. We've found a way to create human insulin. We take the insulin gene out of a cell, and we insert that with enzymes. We cut the gene out of the DNA, the insulin gene, and we insert it into bacteria plasmid. Plasmid is a circular DNA. Now remember, bacteria's DNA is just like ours. It's A, C, T's, and G's. That plasmid is going to be read by a ribosome. They're just like the ones we have. All right, and it doesn't know. It's just the ribosome is going to read that code. And guess what? That bacteria is actually going to produce insulin. And of course, you got to get that bacteria, since it's asexual and makes identical copies of itself, every single bacteria produced from that one that you got. All you need is one. And then you can reproduce it over and over and over, and you can have tons and tons of bacteria producing lots and lots of human insulin. Pretty cool. How do they do it? You know, here's one way. Sometimes they use a virus as a vector, which is going to bring that DNA. And remember, viruses go into the nucleus and kind of hijack the genetic machinery, and that's one way they do it. All right. We've been able to actually insert glowing genes from other organisms that have the glowing gene. There are organisms on the planet that glow, and they stuck it into cats. You got cats that glow. Here, researchers took skin cells from a Turkish Angora female cat and used a virus to insert genetic instructions for making red fluorescent protein. Then they put the gene-altered nuclei into eggs for cloning. All right, so cloning and genetic engineering all go together, and they put the embryos into the donor cat, making the cat surrogate mothers for their own clones. Jellyfish. Now, once again, here we go. What's the good and the bad about all the knowledge we have about the human genome? It's great, right? We can find out if we have a disease or not. And all these on here, this is just the X chromosome. These are diseases, okay, that they're showing on this list. It doesn't mean we have them. This means these are some sort of bad version or mutant version of the normal genes we're supposed to have. And this is just some of them. There's thousands of genes on a chromosome. What's great? I mean, it's great. I'm going to find out if I'm going to have this or that. But, do we always want to know that? Do we want health insurance companies to know that we're going to be sick in a few years and they say no to us? Or you can't get a job because someone doesn't want to hire you because they think you're going to get sick based on your DNA? So it's great having all this knowledge, and genetics is wonderful, and hopefully it's put to good use. But who knows? All right. Well, I hope this little video has helped you out and is a good thing to go back to. It's basically just touching a little bit, but the rest of it in class will explain a lot more and do some activities on it to help us better understand how our DNA actually becomes who we are and how we can actually mess around with it. All right. Have a great night, guys.